In this part, we are going to do the phenotype score calculation, and we use the GSVA library to do this calculation. That's why here we have to upload the library GSVA and the GSVA base, these two libraries, to the R environment. Load gene sets. This is a gene set that contains a lot of gene sets. Each gene set is composed by multiple genes. For example, the hallmark inflammatory response is composed by a number of genes. You can see here the interleukin 1A. That's a gene. It's a gene that is important in the inflammatory response. So one way that you can understand this part is imagining that we need to access the expression of phenotypes in the tumors. And one way that we have to access those values is by calculating how much of certain phenotypes we have in the tumor. So for example, a tumor can have a lot of T-cell inflammation, right? These T-cells are cells that can penetrate in the tumor microenvironment. And to put it in a very simple way, the more immunological cells, the more T cells there are in the tumor microenvironment, the better prognosis that patient is going to have. Just putting it in a very simple way, because this is a much more complicated mechanism. But what we are trying to do here is we are trying to access how much of a phenotype a tumor has. And I gave you the example of how much a hallmark inflammatory response a tumor can have. All right, so we load the gene sets, and I had to reinstall R and GSVA this morning, so that's why this is deprecated. And the new way that I can calculate the scores is it's using new parameters. I think that I have this R2 GSC 62564 underscore matrix here from the previous video. Yes, that's there. So we only need to upload the gene sets now. Okay, let me think a little bit how I'm going to upload this gene set to the environment. And so I'll have to make this data set available in the data folder. It's not there yet, but I'll upload it. I'm just thinking again about the path. So I'm going to copy this line, but if I throw it here, it's going to complain. First of all, it's complaining that it cannot find this function, the get, I think it's get gene matrix function, because the gene matrix is again the gene set, okay? And I'm trying to access in my mind where this is stored in my computer system. What I'm gonna do is I'm going to access that directly from I'm going to use what's called absolute path, and I'm gonna tell you how to do that in a minute. But I'm just realizing that if this function is not in the R environment, it is because I have not ran these two libraries yet. I have not uploaded the GSVA library. one of these two libraries, I should say, not the two of them, and the GSVA base library. So I'm gonna load these libraries, and I'm going to run this command again. Now it's not complaining. It doesn't seem like it is complaining that it cannot load the get gene matrix anymore. What I can, but seems like it's finding somewhere, but I'm going to try to use the absolute path anyways, and I'm gonna see what's gonna happen. So I'm gonna go, this is in a more desktop, Poliano analysis, and then cfDNA. Okay, so I'm going to see if it's, okay, so apparently it went through. So maybe it's just this is just a warning. It ran anyways. So as I said, this is deprecated. I do not calculate the enrichment scores like that anymore. I'm going to use this part here, estimating the GSVA enrichment scores in this new way. So you can see here that I'm going to use the matrix, the gene expression matrix that we constructed from the previous video, and then the gene set list that we just constructed now, we uploaded from my local machine here. So I'm going to copy this. First of all, we need to construct we need to construct this GSVA parameter, where we are just using, so you're using the GSVA parameter function and the gene expression matrix and the list, the gene set. All right, and now we apply the GSVA function. I hope that it's going, okay. It's running. It finished. So this is this, it should be the same output. And now finally, I am going to merge the GSVA, okay, the GSVA object with the gene expression object, right? This matrix was the one that we constructed in the previous video, and then this one is the one that we just constructed. So now we can do this. R bind means row bind, which means I'm binding the first object and the second object by their rows. So I hit enter, and let's take a look at I'm going to do the head and then this object n equals 5. Let's look at the first 5 lines. So you can see here that we have so each column now is the patient. Now remember, from the previous data frame, we also had the patients as columns, and the lines are the gene expression or the gene set. So each one of these is a gene set, okay? So this PCA HR, let me see if it is in here. Yeah, it's the first one, it's sig pos. Yeah, seek pause. So each one of these lines that you see here as the lines in the data frame, they're coming from this object, it's called cfDNA underscore genes underscore PCA. And all of what this is the gene set. Okay. And so there you go. We have just constructed the data frame. And what this data frame is, it is composed of the patient IDs as the columns and the gene expressions or the gene sets as the rows, okay? And that's it. So the next part here, I'm not going to do this because this is using the deprecated data that I just explained a little bit, but the object in the next notebook, the object is going to have this name. So for the next video, the data frame is going to be the data frame is also going to be composed of the patients in the columns and then the gene sets or the genes in the rows. That's it for today. Thank you for watching.